Our third practical focuses on exploring osmosis and turgor pressure using dialysis tubing. The procedure begins by taking a 20 cm length of dialysis tubing that has been pre-soaked in water. Securely knot one end of the tubing to prevent any liquid from escaping. Next, using a plastic syringe, inject 3 cc of concentrated sugar solution into the tubing. Once the solution is inside, tie the open end of the tubing securely. At this point, the tubing will be quite flexible. After sealing the tubing, place it in a test tube filled with water and allow it to sit for 30 to 45 minutes. During this time, osmosis will occur. Finally, after the specified period, remove the dialysis tubing from the water and observe any changes in its appearance and texture. This will allow you to assess the effects of osmosis and turgor pressure within the tubing. As a result, the tubing will experience a visible change, becoming firm and swollen because of the solution inside. So, here's the interpretation of what's happening. The change occurs because the dialysis tubing is partially permeable, which means it lets water diffuse into it. The solution inside the tubing actually has fewer free water molecules than the water outside, so water moves in, causing the tubing to swell and the pressure inside to rise. This is essentially a simplified version of what happens in a turgid plant cell. In this analogy, the sugar solution inside the tubing represents the cell sap, and the dialysis tubing acts like both the plant cell's membrane and its cell wall,